Welcome to Genetics in the NICU, a guide to genetic testing. In this educational video, we will review some core concepts of genetics and then specifically focus on frequently ordered genetic testing in the NICU setting. The NICU setting is unique in many ways and has a high concentration of patients with genetic disease. NICU nurses are central to the care of these neonates and advocates for both patients and families. DNA consists of chemical base pairs. A pairs with T and G pairs with C. Together, these base pairs form a double helix structure, which is in the nucleus of every cell. Known regions in the DNA and what their function or production is are what are considered to be genes. It is estimated there's about 20,000 genes in the human genome, but more continue to be discovered. All of this genetic material is wrapped and condensed very tightly into chromosomes. In total, all of the DNA material is condensed into 23 pairs of chromosomes, 23 from the sperm and 23 from the egg. 46 total. Of the 46, 44 are autosomes and 2 are sex chromosomes, either 2 X's or an X and a Y. The structure of the genetic material is important to keep in the background as we go through each method of genetic testing. Our genetic material is passed on from generation to generation from the point of conception. The sperm has 23 chromosomes, and the egg has 23 chromosomes. After conception, the cells have the typical 46 chromosomes. These cells then divide exponentially, containing the genetic material half from the sperm and half from the egg. An autosomal dominant condition is where there is a change in one of the two copies of a gene, leading to a genetic condition. With a parent who has an autosomal dominant condition, there is a 50% chance with each conception that the child would inherit the non-working copy. An autosomal recessive condition is where there is a change in both copies of a gene, leading to a genetic condition, with parents who are carriers of the same autosomal recessive condition, meaning they have one copy altered but the other copy without a variant, so they themselves are unaffected. There is a 25% chance with each conception that the child would inherit each of the parent's non-working copy of the gene. X-linked conditions are where the affected or non-working gene is specifically on the X chromosome. Males have a 50-50 chance of which X chromosome they will inherit from their mother, and they will inherit the Y chromosome from their father. We tend to see males with X-linked disorders affected more severely because they don't have that backup X to compensate like females do. Depending on the specific condition, carrier females may have a variable expression of the condition, or they may be unaffected. The possibilities in their children are a 50-50 chance of having an affected male, and 50-50 chance of having a daughter who is a carrier of that non-working copy of the gene on the X chromosome. A multifactorial inheritance pattern includes conditions that have a genetic component to the disorder, but also environmental factors that affect the expression and development of the disorder. Common multifactorial inheritance conditions in the NICU include non-syndromic cleft lip and palate, as well as pyloric stenosis. Family history, physical assessment, identification of dysmorphic features, and overall clinical presentation are huge parts of the genetic diagnostic process, and genetic testing is one part of that. All parts are necessary to include when thinking about what type of testing can have the best chance of finding an answer. So, what are the different types of genetic testing in the NICU? Some you may see ordered all the time, some less frequently. Certain testing methods have better yield in a specific situation. Each test has its limitations, even when going super broad. I'll go through common testing methods and the benefits and limitations of each. Chromosome analysis, such as a karyotype or a microarray, are like a general overview and scan of the chromosomes. The completed picture of the chromosomes organized into pairs is the karyotype. Here are two examples of a karyotype. A male with 46 chromosomes, you can see the chromosome pairs numbered 1 through 22, and then the two sex chromosomes, for a total of 46, which is the typical number of chromosomes a human has. On the right is an example of a female karyotype, the autosome pairs numbered, and the two X chromosomes. Here is an example of a karyotype with an extra chromosome, specifically an extra chromosome 13. This is what a karyotype looks like for an individual with trisomy 13, or Patau syndrome. As you can see with this example, a karyotype can identify full extra or missing chromosomes. It can also identify other forms, for example, if a piece of chromosome is translocated onto another. A karyotype can also identify large missing deletions or duplications of chromosomes. For example, if a large segment of chromosome 1 was missing, the karyotype would be able to identify that missing area where it would be expected. However, it couldn't detect if there was a change on the single gene level of chromosome 17. This family could have a mutation in the BRCA gene, which is on chromosome 17, but the karyotype is too zoomed out to detect that. On to a microarray. This is another testing method within the chromosomal studies category, but is a more zoomed-in look of the chromosomes that can identify smaller extra or missing pieces when compared to a karyotype. The microarray basically scans through each chromosome looking for changes in copy number, meaning are there an expected 2 copies of this general region of the chromosome? It basically uses probes that are matched with known areas to the chromosome so it can do a quick count, scanning through detecting if 2 copies are present, and creates this plot of points. You can see on this microarray, where the orange circle and arrow are, the plots dip down to a -1 section. This indicates that in this region on chromosome 7 there's 1 less copy than expected, so there is a deletion in this region. It doesn't say exactly what is in that region and what exact genes were deleted, but it shows that from one point to another, this section on chromosome 7 has a less than expected amount of genetic material. The other arrow is pointing to a duplication, where the section of probes are raised which indicates a duplicated region in this location on chromosome 11. A microarray can detect small chromosomal deletions and duplications, a change in overall number of copies. Depending on what probes the labs use, it can be a little more complex than this and show if someone is heterozygous or homozygous in this region. It can't identify balanced translocations, where one piece of a chromosome is moved and stuck to another with the same overall amount of genetic material, and is still not a high enough resolution to identify a change at the level of a single gene. So what testing method can identify a change at the level of a single gene? This would be sequencing. Going back to our basics of DNA structure, where the base pairs match up with each other, gene sequencing reads through the base pairs looking for spelling changes that may alter the way the gene functions or where the gene functions. A single or multi-gene panel looks in the region where a known gene is located that may be related to how the patient is presenting. A benefit to a single or multi-gene panel is how close it is looking at a specific gene. Compared to a chromosome analysis where it's really zoomed out, this is looking in a known area really closely for any possible spelling change. If the clinician is confident for a specific genetic condition, or if there is a known family mutation, just a single gene might be sequenced. An example of a single gene would be to only look at the CFTR gene in a family with a known history of cystic fibrosis. If it's less clear, or there are a collection of symptoms where there could be multiple genes that could be related to the underlying etiology, a multi-gene panel may be ordered. Some of these panels can get really big, like over 700 genes. But for context, think back to how there's an estimated 20,000 genes, so even a really big panel isn't truly capturing all possibilities. Examples of a multi-gene panel include a large panel for cleft lip and palate, or for congenital skeletal disorders. Expanding on large multi-gene panels, whole exome sequencing is the next most broad method of genetic testing. But what is the exome? Thinking back to the basics of all our double-stranded DNA, the genome is all of our genetic material in total, including regions that code for proteins and the regions that are in between. The exome is only the coding regions, or just the regions that code for proteins within the genome. Whole exome sequencing combs through these coding regions and looks for spelling changes. The exome is only about 1.5% of the total genome, which may seem tiny. But these regions are very informative because ultimately protein leads to function. It makes sense to look at areas that if a protein was altered or missing, it would affect function, which could lead to the presenting clinical symptoms. The lab reports back the results, if any changes in the DNA sequence were found, and the interpretation of the result. When looking through all this genetic material, It helps the lab when there is clear phenotypic information, meaning what features the patient is presenting with. This provides information that are like context clues to aid in interpretation. This is why careful and detailed physical assessments are very important. If parents are available for testing, a whole exome is often sent as a trio, meaning it includes samples from the two biological parents of the patient. This can help contextualize the findings in the patient. If the same variant was seen in one of the parents, or it's a new variant only in the patient. In variations in DNA sequence that are rare, or little literature is available, this can really help. If one of the parents have the same change, but does not have the same clinical presentation, it makes it less likely that that change would be responsible for how the patient is presenting. Moving on to whole genome sequencing. This is sequencing of all the genetic material in its entirety, including both the coding and the non-coding regions. Genome sequencing is the largest breadth of testing currently available. It is indicated if other methods have resulted negative and a wider net is needed, or if there is an unclear phenotype that doesn't point to a direction of genetic disease, but there are features present that indicate a potential genetic etiology for the presenting condition. Although genome sequencing is the most comprehensive form of genetic testing, it has its limitations. It can miss large deletions or rearrangements, which may be better detected from a chromosomal study. The vast amount of data that results from looking through all of an individual's genetic material can be difficult to correlate clinically. A variation, also described as a mutation, was identified through the genetic testing. This variation is known to be associated with genetic disease, and there is strong evidence in the literature or in the lab that this change would be pathogenic or disease-causing. With a negative test result, this means that the test did not identify a genetic change. However, the negative result is considered with the limitations of the testing technology that was used. Further testing may or may not be indicated following a genetic testing negative, depending how it is correlated clinically with the patient's presentation. This is often the least straightforward type of result for patients and clinicians alike. A variant of uncertain significance is not uncommon. It is when a variant was identified, although its relation to disease is uncertain. A variant of uncertain significance may require additional testing, further literature review, or may not be clinically significant. Variants of uncertain significance are definitely challenging to correlate clinically. The labs that perform genetic testing maintain this data, and as more information becomes available, they will periodically revisit and may change the classification of a VUS. Secondary findings are when a variation was identified through genetic testing, but the variation that was found isn't related to the original indication for genetic testing. Secondary findings often result from exome or genome sequencing, but can also be found through a karyotype or microarray. For example, a son had a whole exome trio. Parents opted in to secondary findings during the consent process. A variant in the BRCA gene was found in both mom and son. This has implications for both of their healthcare, as it's an adult-onset cancer predisposition gene, and so mom may want to seek genetic counseling herself to discuss recommended screening and prevention. For the son, this may have been found but is unrelated to his current condition and the reason genetic testing was sent in the first place. But since it was found and has known implications, he should retain these records of this for when he's older. The American College of Medical Genetics has a list of reportable secondary findings, meaning they won't release just any variant that's found in any gene, only a pathogenic variant in a gene that would cause a condition where medical intervention could affect health outcome, like in the example with BRCA and its risks associated with breast cancer. Incidental findings are similar to secondary findings in that they are a result identified that wasn't the reason the test was sent, however differ in that this result, if revealed, may not have a clear medical benefit versus its potential harm. A good example of an incidental finding would be discovering misattributed parentage, such as non-paternity or non-maternity. A common question is, will the genetic testing reveal non-paternity or non-maternity to the parents if it's found through genetic testing of the baby? The answer isn't exactly a yes or no, and is discussed frequently as an ethical consideration in genetic testing. Ultimately, the stance of the American College of Medical Genetics is that the possibility of identifying misattributed parentage through genetic testing should be discussed as a part of pre-test counseling by the genetics health professional. If a trio is submitted and not all samples are biologically related, the sample may then be run by the lab as just a duo. The lab would notify the ordering provider of this. The genetics health professional, depending on the situation, can reveal that the sample was unable to be run as a trio, but it doesn't have to be stated because of suspected non-paternity, for example. There's other reasons a sample would not be run. However, some argue ethically that this information should be disclosed. At the end of the day, exome or genome sequencing is not validated for paternity testing, but it can be identified or suspected. Often the way this situation is handled is it is discussed as a possible finding in pre-test counseling, and if this is revealed to the ordering provider may choose to disclose if deemed medically beneficial. It can also be discussed with the family to seek methods of paternity testing that are validated for that specific indication if they are interested. Again, this is an ethically complex situation that does arise fairly frequently with genetic testing. It is often dealt with on a case-by-case basis, but a non-paternity result wouldn't be automatically released to the parents in that plane of language. Moving on to genetics in a public health space. Newborn screening is a public health initiative to screen all newborns for select, well-understood conditions that have early onset, where early intervention can improve health outcomes, where medical treatments are available, and there is availability of a confirmatory test. The conditions included in newborn screening differ from state to state. In the state of California, all newborns are screened. Parents or guardians are able to refuse for religious reasons. In the NICU setting, there are certain considerations because of the patient population. In a critically ill neonate in the NICU, metabolic instability, blood transfusions, TPN, and prematurity can all affect newborn screening. Repeat screening may be indicated. The results are reported against a reference range and reported out, depending on the situation of the newborn. If they are discharged home before results return, the designated pediatrician will follow up. An out-of-range result triggers need for follow-up and potential confirmatory testing. As newborn screening is a screening tool, there is the potential for false positives. The screening is designed to be sensitive, so false positives are not uncommon. Confirmatory testing varies depending on what the condition was that screened positive. It may be further biochemical testing, enzyme levels, algorithms for intervention, or confirmation with molecular genetic testing. Cystic fibrosis, for example, if screened positive on newborn screening, reflexes to molecular sequencing of the CFTR gene for common mutations. This is not done for all of the conditions included in California newborn screening, which is why consultation with genetics professionals may be indicated for further genetic testing. Newborn screening has had great successes, but also has its limitations. Primarily, it is screening, not diagnostic, and is meant to identify pre-symptomatic and at-risk newborns, but still requires follow-up and confirmatory testing. False positive rates, or false negatives, are a possibility. Results of newborn screening may not return quickly enough in a rapidly deteriorating neonate. There are confounding variables depending on the situation of the newborn, including TPN, transfusions, and maternal conditions. There are some current ongoing studies performing genetic sequencing as newborn screening and moving away from solely biochemical and metabolic analytes. These are some current studies seeking to determine if genetic sequencing as a screening tool elicits more comprehensive results and if there is utility compared to current screening methods. This may be a direction where newborn screening is headed in the future.